So first of all, welcome everybody today. My name is Sarah Hudson, so I'll be chair for today's lecture. Thank you for coming. First of all, just a little bit of housekeeping. Can you make sure your phones are on silent? And also just to note, the exits are just up at the back of the theatre and down at the front to the sides. So now it gives me great pleasure to ask our president of the University of Limerick, Professor Fitzgerald, to introduce our second Bernal Distinguished Lecturer. Thank you.
Thank you, everybody. I'm delighted to be at the second Bernal lecture that I've been at. I think we've only had two, is that correct? Yeah. And these are very important lectures because they celebrate the work of major scientists and engineers, like our speaker today. But importantly, they are lectures that are open to the public, and they're designed to tell the story of how research impacts on our lives, and in particular with Bob Langer on the development of new treatments. I know that Sarah was the person who brought Bob here, and Sarah has in the past had worked with Bob and before she came to UL. So I should say this is an extraordinary day for the institution to have somebody of his stature, and we're very grateful to Sarah for organising that. Our speaker today is an extraordinary individual, and I was reading his bio and reading some of the comments that people have made about him over the years. As you can see, he's the David H. Koch Institute professor at MIT. And MIT has only a limited number of institutes, so it's quite an honor to be one of the institute chairs. He's the most cited engineer in history. Most people's H-index sort of at best hover around their age, so unless he's 233, he's done an extraordinary job in the impact that his research has had over the years. He's written nearly 1,400 articles, and he has 1,130 issued and pending patents worldwide. And many of these patents have been sub-licensed to companies for the development of products. He's also been awarded some of the highest awards in science and engineering in the United States. He was the United States Medal of Science awardee, and he's also been awarded the National Medal for Technology Innovation, and I understand there's only 4 people who received both of those, so that's, again, an extraordinary achievement. His lecture today runs through a lifetime of work. He has always promoted an interdisciplinary approach to work involving scientists, medics, computer science, mathematics, engineering, and I think this is very similar to the approach that we've taken here at the Bernal Institute. Indeed, Bernal Institute opened just a little less than a year ago now, and by the Taoiseach, I was here that day. It was a terrific day, and now it has 60 academics and a large number of PhD students and postdocs, really following a similar path in bringing people from different disciplines together to solve problems, and particularly the problems that industry is facing, as well as the basic science problems. Bernal Institute, I think, will continue to build on the centres that have been established here, and I hope in the next few years we'll see other centres here and other Bernal professors setting up their laboratories here. It truly is a fantastic institution, and I think we can look forward to some extraordinary things happening today. Maybe someday one of our Bernal people will have the H-index of 233 also. I'm delighted that Bob is here. I hope that you engage after the lecture in a vibrant Q&A, and I hope that he enjoys his stay here on the campus. So would you please welcome our speaker for today, Bob Langle.
Thank you very, very much. Well, thank you very much. It's really an honor and pleasure to be here. You know, what I thought I'd try to do today is tell you a little bit about my own career and how a chemical engineer like myself got involved in the biomedical area, and then go over some specific projects we've been working on, starting long ago on drug delivery systems, as was mentioned, and ending on new ways to create actual tissues and organs. So I'm going to start at the beginning. I was a graduate student, like many of the people here at MIT, in chemical engineering. I'm a few years older, so I got my degree in the 1970s. And when I got my degree in chemical engineering, there was this sort of gas shortage, and the price of gas kept going up and up. And in Cambridge, where I lived, if you had a car, not only did you see the prices go up, but you actually had to wait in line at a gas station for 2 hours to get your tank filled up. But the consequence of that is that if you were a chemical engineer, you got a lot of job offers. And pretty much every one of my classmates at MIT went to the oil industry. And I thought that's what I should do too, because that's what everybody else did. And so I actually applied to different oil companies, and I actually got 20 job offers, 4 from Exxon alone. And I wasn't that good, but they just had so many openings. One of them made quite an impression on me. I remember going to Exxon in Louisiana, and one of the engineers said to me, he said, you know, if you could just increase the yield of this one petrochemical by 0.1%, he said, that would be fantastic. He said, that would be worth billions of dollars, you know, 0.1%. And I remember flying back to Boston that night thinking to myself that I really didn't want to do that. And it was a great lesson for me to learn what I didn't want to do. Made me think, well, what did I want to do, rather than just follow all my colleagues? And one of the things I did when I was a graduate student at MIT is I helped start a school for poor high school students. And I got very involved in creating new chemistry curricula and new math curricula to try to make those subjects more interesting. One day I saw an ad at the City College of New York in the United States to be an assistant professor to develop new chemistry curricula. And I thought to myself, that's great. I wrote them a letter applying for a job, but they didn't write me back. But I really liked the idea. I found all the ads I could to be assistant professor to develop new chemistry curricula. And I wrote to about 40 colleges, and actually not one of them wrote me back. So that wasn't going so well. So I started to think, well, what other way could I use my chemical engineering education to help people? And I thought about medicine. So I wrote to a lot of hospitals and medical schools, and they didn't write me back either. But then one day I walked into the lab, and one of the postdocs said to me, he said, Bob, he said, there's this surgeon in Boston named Judah Folkman. And he said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. But anyhow, I'm going to and Dr. Folkman was actually kind enough to offer me a job. And I'm going to start by showing you a picture from the New York Times of Dr. Folkman's vision of how tumors grow. So this was a totally different area. He had this idea that tumors grew like this. And actually, this is an engineering problem in a sense. His idea is that somehow the tumor cell would become abnormal. It would then grow to this three-dimensional mass. And yet, he said, it would not get larger than about 1 millimeter cubed because it ran into a nutrition problem. Cells in the center would die because they couldn't get nutrients or get rid of waste. So you wouldn't get basically more than 1 million cells or a millimeter cube diameter, except for the fact that he noticed and said that the tumor creates new blood vessels. And his hypothesis was that it actually made a chemical signal, which he called TAF, or tumor angiogenesis factor, that causes the surrounding blood vessels, which are normally quiescent, don't do anything, to start growing right to the tumor. And that, he said, would cause a second phase of growth, a vascular phase, because it solves the nutrition problem. And because the blood vessels go right into the tumor. And the tumor now can get bigger and bigger and bigger, and it can actually spread through those blood vessels. That's a process called metastasis. That's what often kills people. And his idea was that somehow if you could stop this from happening, achieve anti-angiogenesis, maybe that would be a whole new way to think about treating cancer. I thought this sounded fascinating. I had no idea. This was an incredibly controversial idea. Most people totally disagreed with Dr. Folkman, but I thought it was really neat. At any rate, when I came to the lab, he said, well, I'd like you to prove this theory. And in so doing, could we actually isolate for the first time a substance that would stop blood vessels from growing? So how do you think about a problem like that? Well, being an engineer, I kind of broke it down into two parts. First, where could you ever think about finding a substance that could stop blood vessels from growing? Well, one of the things we thought about was cartilage, which is in your nose or your knee. Cartilage doesn't have blood vessels. We had some small rabbits in the lab. I got some of their cartilage, but I couldn't get that much. One of the things you learn in chemical engineering is scale up. So I went to cows. I found a slaughterhouse in Cambridge, and I got a cow bone. Still, one bone, that's not much cartilage either. So what I did is I found out, where do all the bones, all the cow bones in the New England area go? And it turns out, as I tracked this down, they went to these meat packing places in South Boston. And I made an arrangement with them to get all their bones. I'd go down there 4 times a week, bring their bones back, scrape the meat off, in fact, and then I would create something like this. This is actually one of those cow bones. It doesn't come like that. Normally there's meat on this, and I would scrape it off with what's called a periosteal elevator. I'd probably spend about 40 or 50 hours a week just doing this. I got really good at it. And then I would slice this piece of cartilage off, and then I would put it through various extraction procedures with chemicals like guanidinium hydrochloride, and then I'd put it through different purification columns. And at the end of a couple of years, I had maybe 50 to 100 different what I'll call fractions. That then brings up the second problem. How would you test whether it was stopping blood vessels? And if you look back at the history of medicine, some of the things that you see are that almost what's always rate limiting in terms of trying to identify a factor and if it's working is what's called a bioassay. And there wasn't one. And it's not easy to have a bioassay to study new blood vessels because they grow relatively slowly. What we thought about, though, was maybe one way to do this, to create a bioassay, is to use the rabbit eye. This is going to be from a paper we published in Science. I'll come back to this in 1976. So our idea was that you could put certain types of tumors in, like a V2 carcinoma. And what I'll show you soon is that tumor will, over about a 10-week period, cause blood vessels to grow from the edge of the cornea to the tumor. And you could actually quantitate that with an ophthalmic microscope with the rabbit. And the rabbits have big eyes. So basically, we'd put the tumor in, but then we'd need a polymer that could, A, be inert in the eye, not cause damage, and also had the ability to release molecules of any size. The molecules in cartilage were often large, like proteins and proteoglycans. So we needed it to be able to release these large molecules over this 10-week period, let's say, so that we could study it. Now, that had never been done before. And Dr. Folkman was actually on the board of the one company in the world, a company called Alza, that had ever done any work actually on delivering molecules at all. And they were very tiny molecules. And he asked them, could they help us? And they said, well, no. They said, large molecules can't slowly diffuse through solid polymers any more than any of us could walk through a wall. In fact, the literature said the same thing. The use of polymer matrices for slow-release systems has been virtually restricted to small molecules. Now, the only thing I really had going for me is I hadn't read any of this. So I tried anyhow. I spent 2 years in the laboratory experimenting with different techniques. I actually figured out over 200 different ways to get this to not work. But eventually, I figured out a way to make little microspheres like this. And what we would do is we would use basically different hydrophobic polymers. They could be non-degradable polymers like ethylene vinyl acetate copolymer, or degradable polymers like lactic glycolic acid copolymer. We would dissolve them in a organic solvent, add drug to it, and often we do these at very low temperatures, and we'd get these little microspheres, and here's one cut in half. And then we published, actually in Nature now, for the first time, that you could release these molecules for over 100 days. These are different enzymes or enzyme inhibitors, all being released for long periods of time. And in these early days when we first did this, as you can see, the release rates are not constant. But later on, using some mathematical models, we figured out ways to get them designs to come up with them to be constant. And here we're releasing albumin, which is 68,000 molecular weight for over 50 days. So in 1976, for the first time in my life, I got asked to give a talk at a major scientific meeting. And I was very, very nervous about this. In fact, the only talk that I'd ever given before this at all was in 8th grade. And that didn't go real well. In 8th grade, I remember I had to give this minute-and-a-half speech to my class. And I remember writing it out the night before and standing in front of my parents' mirror for 3 hours, reciting it over and over again. And then the next day came, and I remember getting up in front of my 8th grade class, and I started reciting it. And for the first minute and 2 seconds, I did okay. And then I could not remember the next word. And I just stood up there absolutely frozen for another 60 seconds, saying absolutely nothing, until my 8th grade teacher finally told me to sit down and gave me a not very good grade. It was an F. And so now, many years later, I had to give this talk. I'm now 27 years old at the time, and I had to give this talk to this very distinguished group of polymer chemists and engineers. So this time I stopped work about 2 weeks in advance. I kept reciting the talk into a tape recorder. They didn't have much video stuff then. And I got up and I gave the talk to this really very distinguished group of scientists and engineers in Michigan. And at the end of the talk, I thought I did all right. I didn't forget too much what I was going to say. I didn't stammer too much. And I thought when I was done with this talk then that all these older chemists and engineers, being nice people, would want to encourage me, this young guy. And when I got done, I stepped off the podium, and a whole bunch of them came up to me, and they said, we don't believe anything you just said. They said, just like you heard before, that they said large molecules can't slowly diffuse through solid polymers, and also we were using organic solvents. They said that'll destroy whatever you're putting in. Actually, things kind of went downhill from there, if that was possible. But they did. Actually, what happened was, first I tried to get research grants to support this work. And my first 9, I got totally rejected. And then, I really liked being a postdoc, but a lot of my friends said, probably it's not a good idea to be a postdoc your whole life. So I started applying for assistant professorships, faculty positions, at different chemical engineering departments, which was my discipline. But not a single chemical engineering department in the United States would hire me. They thought this kind of bio stuff, which might seem common today, nobody did that then. They said that this doesn't seem to be chemical engineering. So I got 0 job offers. Eventually, I ended up being at a nutrition department. And that would have been okay, except the only issue there was that a year after I joined the department head, who was kind of what I would think of as a benevolent dictator. In other words, he wanted to hire me, but he didn't ask anybody else in the department. And then he actually left. So a lot of the other faculty members decided to give me advice. And their advice is I should start looking for another job. Let me actually back that up. A couple years ago, I got this big biotechnology award, and they asked Michael Marletta, who was one of my colleagues at the time. Mike has actually had a very distinguished career. He's in the National Academy of Sciences. He was at one time head of Scripps. At any rate, here he's just telling a story about what it's like to be at MIT in the around 1980. And he said one evening he went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for polymeric drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. And Mike said he thought he was in a Fellini movie. Well, so like I say, the start wasn't so auspicious. Fortunately, after a few years, people started using some of our principles, and I got to keep the job. At any rate, one of the questions that kept coming up, though, was how could this possibly work? Because people started now by reproducing it. You heard about the H-index and citation. So people kept starting to reproduce it, but it was unclear to people how it worked, and people still were skeptical. I had a graduate student, Rajan Bawa, and I asked him to look into this, and we did a lot of microscopy. And one of the things that we did is we'd cut thin sections through some of these polymers, like ethylene vinyl acetate copolymer. And if you do a 5-micron thin section, what you see is if you do this before any release takes place, or you don't put any drug in it, it looks like this. It's a sheet. So molecules 300 molecular weight or greater can't get through. But if you put a drug in, and now we'll put a reddish molecule in just to visualize it, you see, before any release has taken place, what we call a phase separation. The polymer is here, the drug is here. If you then release this, say for a year, and then you cut a thin section, what you see, in sharp contrast to what you saw 2 slides ago, are all these pores. They didn't exist before. They're made by the way we did this. And it turns out that these pores are large enough so that molecules even millions of molecular weight can get through. But it also turns out if you do serial sectioning or scanning electron microscopy and image analysis, that these pores are actually interconnected. There are tight constrictions between the pores. And the pores are incredibly winding and tortuous. So it takes a very, very long time for the molecules to get through. One way I sometimes explain this, if any of you have ever been to my hometown of Boston, it's kind of like driving a car through Boston. Boston and these systems have what we call in chemical engineering high tortuosity. And over the years, quite a few of our students, like Ron Siegel and Rajan Bawa, Larry Brown, and others, have actually identified ways to mathematically model these systems, develop actually rules for controlling release. So now you can actually design these systems to last anywhere from a day to many years, or any time in between. Now, since we can do this, we could try to go back and solve the angiogenesis problem. So remember, this was the assay that we wanted to develop, tumor in the polymer. And now I'm going to show you something very graphic. And if anybody doesn't like the sight of blood, don't look. But there's no other way to show this. What I'm going to show you is where we actually, and this is from this paper in Science we wrote in 1976, where we actually put tumors in the eye, and we followed probably 2,000 rabbit eyes. And basically, we tested all the different fractions, probably an N of 15 or so for each fraction, as well as blank controls. And I should say that what we're looking at are blood vessels about 10 weeks later. So again, just as a warning, don't look if you don't like the sight of blood. But here's what it looks like from this Science paper. So if you did not have the cartilage-derived inhibitor, after about 10 weeks, what you see is a sheet of blood vessels. You can just see the tip of the polymer. The polymer's here. Here's the part of the tumor. If you kept this animal alive, what you'd see is just 3 weeks later, the tumor would be 3-dimensional, out of the orbit of the eye. It would actually kill. We sacrificed them before that. In contrast, when you put the polymer in with the inhibitor, the cartilage-derived inhibitor, here again is the tumor. Here's the polymer. Notice how the blood vessels are lower. They're much sparser, and they actually totally avoid the polymer. So this was quite reproducible. This happened every single time. And in fact, from a tumor standpoint, 100% of these tumors, and we did thousands, grew 3-dimensional. Here, 60% of the time they did not, which was unprecedented. So we published this in Science. That was 1976. But and this often happens in medicine. Medicine's slow, and we didn't have a lot of funding. So it took another 28 years from the Science paper before the first angiogenesis inhibitor was approved for clinical use. And that probably cost billions of dollars. That was actually done through the work of Genentech and then later many other companies. On this next slide, though, I'm going to show you where it stands today. And this may be hard to see in the back of the room, but what's striking is starting in 2004, 28 years after this first paper, what you see is one after the next angiogenesis inhibitor approved by FDA. Avastin, which was the Genentech drug, still is, has been the #1 or 2 largest selling biotech drug in history. It's still today the 4th largest selling drug in the world. And it's been approved for many different types of cancer. There are many other angiogenesis inhibitors approved too. This, by the way, is not even a complete list, but it gives you a sense of the different kinds of cancers that these have been approved for. Not only has these been approved for cancer, but there are very bad eye diseases, macular degeneration, diabetic retinopathy, where you have abnormal vascularization in the back of the eye. And before this, there were no drugs that could treat them that would make people go blind. And now, for the first time, there are drugs like Lucentis, Eylea, which not only could stop those diseases because you prevent the vascularization, in some cases even reverse them. These are also blockbuster drugs, many billions of dollars of sales. And this continues to right now. In April even, Lucentis approved for diabetic retinopathy, which is one of the major complications of diabetes. So right now we've seen all these angiogenesis inhibitors really help millions and millions of people around the world. But it also occurred to me that the drug delivery systems themselves could be useful not only for enabling the isolation of these angiogenesis inhibitors, but in their own right for delivering drugs that are really hard to deliver. And so just as a proof of principle, Larry Brown, another one of our graduate students, took insulin and put it in a little aspirin-sized pellet, put it underneath the skin of diabetic rats, and was able to lower the blood sugar for over 3 months. Dr. Folkman said to me one day, he said, Bob, we should file for a patent. That might seem common today, but in the 1970s, nobody did that in academia. And Children's Hospital, where I was, and a lot of people would say Boston Children's Hospital is the best children's hospital in the world, they had 0 patents. We filed for the first one. 5 years in a row, the patent examiner turned it down. And the lawyer for the hospital said, Bob, you are just wasting the hospital an enormous amount of money. He said, just give up. He said, this is never going to issue. But as you can probably tell by now, I'm pretty stubborn. And I was trying to think, we had to convince the patent examiner to allow this, legally, of course. And and science wasn't working. We kept trying to explain to him about everything. It wasn't working. But as I told you in the beginning, when we first started talking about this, everybody told me this was impossible. It would never work. And I started to think, what if somebody wrote that down? So I did what's called a science citation search, which means I could look back. I did this in 1982. I could look back at our 1976 Nature paper and see who wrote different things about us. And I found some very interesting articles. I'll just show you one. This is from 5 of the top polymer chemists in the world. That they wrote this in 1979. I had no idea they wrote this. But when I found it, I was quite pleased. What they wrote, they're describing this field. They said, generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released by such a technique because of their extremely small permeation rates through polymers. However, Folkman and myself have reported some surprising, that's a really good word for a patent examiner, some surprising results that clearly demonstrate the opposite. So I showed that to our lawyer, and he was very excited. He flew down to Washington, showed it to the patent examiner. And the patent examiner said, I had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from each of these people that they really wrote this. So I wrote each of them, and they were all nice enough to write me back that they really did write it. And we showed that to the examiner, and he allowed this very broad patent. And with that patent, we would license things to different companies. We'd teach different companies. And today, there are many, many products based on this that affect millions and millions of people around the world. I'll just give a couple of examples. If somebody has prostate cancer, endometriosis, there are peptide hormones that people want to use. But they're so big, you can't swallow them because they won't get absorbed. They tried to deliver it through the nose. You couldn't get any in. They inject it, but then it's destroyed almost immediately. So now they put them in little microspheres or rods, and originally they were able to get them to last for a month. Now they last for 6 months, and those have led to whole new treatments. Other examples are drugs for people with mental health diseases, like schizophrenia. This, for example, is a once every couple week system that's used for treating people with schizophrenia. If people don't know what that is, if you ever saw the movie A Beautiful Mind with Russell Crowe, where he saw other people, that's schizophrenia. Millions and millions of people use this. Many others, too. There are peptide hormones that are used for type 2 diabetes, and these also, before this, you wouldn't really use them because they have to be injected every few minutes. I mean, it's just not practical. So now, like I say, there's many of these and many more to come, I mean, that are in various stages of clinical trials. But we always want to do better. And in academia, what I started thinking about, and I got involved in advising companies and helping start companies to do this, because people didn't think that would happen either, but like you can see, it has. But then I started thinking, back on the MIT part, what I started thinking is, well, what's the next big challenge? And the next big challenge is, see, these systems you can inject underneath the skin and subcutaneously, intramuscularly, they will deliver the drugs at a controlled rate for a long time. But what they don't do is they don't target the drug to the cells you want. And I wanted to solve that problem too. And one of the challenges, and here I started thinking about nanotechnology. Could we make little nanoparticles that could circulate around the bloodstream and go to where you want? But there's a big challenge there, because whenever you try to do that, there are other cells, macrophages, that eat things. But we had the idea that maybe what we could do is create a disguise for these nanoparticles where we could add something. Here we made these dye-block copolymers with polyethylene glycol, which is FDA approved on the outside, but that PEG will absorb or attract a lot of water. So in a way, that's going to be a disguise, because normally these macrophages will see a particle and they'll just eat it. But if they just saw water, they see that anyhow, so then maybe they won't eat it. So we published this in Science in 1994, and we've continued to be working on this. I'm not going to explain that much detail, but I just wanted to show you one video that Nova, the TV show, did on what we did a few years ago, just to illustrate where this is all heading. And it really just extends the earlier work we did, but takes it into a new direction to try to make it targeted to cells. Here's the video.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That, in turn, gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
I should point out that a lot of the clinicians I work with tell me it doesn't blow the cell up quite like that. But it gives you the idea. And some of the things, and again, we're in various stages of clinical trials like this and doing still a lot of basic research on this, is that maybe some of the things you think about are delivering small molecules like anti-cancer drugs to a cancer cell. But I think what's particularly exciting about these kinds of approaches, whether they're polymeric nanoparticles or lipid nanoparticles, and we're working on all these things, is what it may enable for the future. Because some of the newer drugs that I think will really change the world are drugs like siRNA. These are ways of shutting down genes. Or messenger RNA, which is giving you new genetic opportunities. In other words, gain of function. These molecules, small molecule might, drug delivery system will make it work a lot better. These molecules, if you don't have a delivery system, you can't do anything. So really figuring out the right nanoparticles, to me, is going to enable some of these. And I think it's moving. I mean, my feeling is 20 or 30 years from now, just like proteins, when we started in 1970s, where the sales were almost nothing. Now it's over $200 billion, affecting billions and billions of lives. Well, right now the sales of these are nothing, but this is in a phase 3 trial. This is in multiple phase 1 trials. I would expect 20 or 30 years from now, particularly if people here and other places solve the delivery problems, ourselves as well, you'll see that this will change the world. I mean, this will really have a giant impact. But delivery is going to be a huge deal. So, and like I say, we publish a lot on this every year, but it's a tough area, but one I think that has enormous opportunities for material scientists and biochemists and so forth. I was going to now shift gears a little bit to another delivery system that I had an idea of on. And one day, I was watching this TV show in the United States about how they were making microchips for the computer industry. And when I watched it, I thought to myself, this would be a great way to make a drug delivery system. Now, again, if you spent 30, 40 years of your life working on drug delivery systems, you might think that about any TV show. But when I saw it, I thought maybe we could make a microchip, but not to deliver, to do electrical things, but to do chemical things. Could we make a chip, cover it with a metal like gold or a platinum alloy, and the drugs are going to be here, and this would actually stay in the body. This is just a cutaway for however long you wanted it to be. But the idea is we could blow individually any cover off we wanted, and the drug could come out. So I worked with Michael Sema, one of my colleagues on this. We had a graduate student, John Santini, and we make these little chips with microfabrication. Here's just an early example of a chip, a top view with 34 wells, bottom view with 34 wells, and this is the smallest United States coin, a dime. Let me just show you how they work. Here's a well, covered in this case with gold, but what you can do is by remote control, you could actually turn on a signal, and you could, with the right voltage and so forth, the cover will come off. And it comes off in nanoseconds. And when the cover comes off, whatever's underneath it can come right out. Here's from another paper in Nature that we wrote with John, our student. And what you see is different amounts and different wells we made, this is in vitro, at different times. You could literally think about this someday as like a pharmacy on a chip. If the drugs are potent enough, you could put any drug you want or any combination of drugs and program them to come out at different times. And as a proof of principle on that, in that same Nature paper, we delivered multiple different model molecules. But what we did in this case is we took this from test tubes. John actually started a company on this, which we helped him, and called Microchips Biotech. And what we did is we went from test tubes to small animals to large animals to actually human beings. And this may sound like space-age medicine, but it's being done. So what was done in the first human clinical trial, this was done in Denmark, is that you put the chips in the human body. You communicate with them over a special radio frequency called the Medical Implant Communication Service Band. It's actually approved by both the FDA and FCC. That's the Federal Communications Commission. You can actually have a special computer code to administer or change the dose if you want. The doctor could do it or you could do it. You also have a bidirectional communications link between the chip and the receiver. Like I say, the receiver could be something like this, or it could be an iPhone type of thing, you know, and basically it tells you everything. Did you take the drug? I have to admit, as I'm getting older, I need this more and more if it was available, and other information too. Well, let me tell you about the first clinical trial. It's 8 patients. Obviously, we want to see if it's safe, but also we wanted to see if it was effective. And again, whenever we proposed these things, people told us it wouldn't work because they say you'll get fibrous encapsulation, drug won't come out. So we said, let's pick a really hard drug, a big drug, like a peptide, parathyroid hormone. It's 39 amino acids. If that comes out, you'd like to think other things will come out. So also we picked it because we felt it could make a big clinical impact. 77% of the women who have osteoporosis, that's the disease you treat with parathyroid hormone, stop taking it. You have to actually inject it once a day. You don't want continuous release. You want pulsatile release, otherwise you'll get bone resorption. So we felt it could really make an impact. So so what was done in the trial is a small office procedure to do the implant. We got the same pharmacokinetics with actually less variability than injections, and these are 3 different measures, calcium and different collagen fragments, of treating it as you do with a regular injection. Here's some real human data. This is the release curves in human beings at day 60, 68, 76, and 84. So it's very reproducible. Here's actually an early level implant. There's a chip. There's a power source in here. There's microelectronics and even a computer program. And finally, we actually imprinted an antenna into this. That's how you communicate with it. And that's how you can do all this. You do get some fibrous encapsulation, but it's pretty minimal, maybe, say, 1/20th of a pacemaker. You do what's called histology, and that's this. You don't see, you do see a little bit of, again, tissue, but no inflammatory cells. So this is really the first example of what we did. But, you know, we always want to take these things further, and part of the challenge is sometimes thinking about what the right applications are. And I had a visit one day from a very successful software entrepreneur. And he and his wife have this foundation. And they like to help people in the third world. And one of the things they were very concerned about was birth control. And actually, I'll just show you a picture in my office. This is me. Here's the software entrepreneur. And it's Bill Gates, in case anybody didn't know. But at any rate, one of the things that they were very interested in is right now in Africa, women, you know, very commonly get abortions. It can kill people. That's not done very well. The birth control systems that exist today, they basically last no more than 5 years. And in particular, what they were interested in is there's no way to change it. In other words, to turn it on or off. Could you ever do that? And so they said, well, could we ever design this? This is actually right from the Gates Foundation website. Could you develop a personal fertility control system with emphasis for use by women living in the developing world countries as a means to effectively plan their families? So the idea, just to at a high level to explain it, is that maybe what we could do is have a chip with 200 wells. Each well is a 1-month supply. And basically, you have a program that has each cover come off each month. That'll take you 16 years and 8 months. And if the woman changes her mind, she presses a button, program goes off. If she changes her mind the other way 2 years later, pushes a button, it goes back on. So these are just examples. Another thing, of course, that I'd love to see happen as time goes on, and we're too early for this, is you could really make a smart chip, put biosensors in the chip, have it detect signals in the body and tell the chip in return how much to deliver. Well, so far what I've done is I've talked to you about how we could take materials that already exist and make them do things they never did before. But I want to spend a few minutes on the materials themselves. One of the things that I became curious about as I was working on this is, how did materials find their way into medicine in the first place? Was it by chemical engineers or chemists that were, say, older than me? Well, as I looked into this, I found out it was almost never true. Almost always, as I looked into this, since I also worked at a hospital, was that the driving force for bringing materials into medicine were medical doctors. And what they always wanted to do, because they urgently wanted to solve the medical problem, is they'd usually go to their house and they'd find some object that kind of resembled the organ or tissue they wanted to fix, and they'd use it in a person. For example, in the case of the artificial heart, this goes back to 50 years, to 1967. And clinicians at the National Institutes of Health wanted to make an artificial heart. And they said, what object has a good flex life like a heart? And they said, well, a lady's girdle. So they took that material and they made the artificial heart out of it. That was 1967. That's 2017 as well. Because once you start down that path from a regulatory standpoint, it's very hard to change. But one of the problems with the artificial heart is when the blood hits the surface of it, the lady's girdle material, it can form a clot. And that clot can go to the patient's brain and they can get a stroke and they may die. But if you think about it, something that's designed to be a lady's girdle, it may not be the optimal blood contacting material. And this problem pervades all of medicine. Just another quick example, breast implants. One of those was actually a mattress stuffing. So I thought, well, why can't we use a totally different paradigm? Why can't we use an engineering approach? Rather than take materials from your house, why couldn't we ask the question, what do we really want in a biomaterial from a chemistry standpoint, biology standpoint, an engineering standpoint, and then synthesize it from first principles? And so we picked an example. The example we picked when we first started were synthetic degradable polymers. And when we started, the only materials that were approved for use that were synthetic and degradable were the polyester sutures. And they dissolve like this. They start like this, they get spongy, they fall apart. And when that happens, it has the potential to have bursts of drug coming out, which would be very bad, say, if you had an anti-cancer drug or insulin. So you could kill. So we said what you really want is something like this, surface erosion, layer by layer erosion. That way you couldn't get this dose dumping. And the challenge, though, of course, is how do we solve that problem? And so what we did is ask different engineering design questions, like, first, if we want to get this kind of erosion, what should cause it? Should it be an enzyme or water? Our thinking was that everybody in this room may have different enzyme levels, but you all have excess water. So what we decided to do was build into the polymers that we were going to synthesize the ability to be specifically degraded by water. And then we would go over other design questions, like what should be the building blocks, what are called the monomers, of these polymers. And they had to be safe, and they had to be, the ability to change release rates. And we'd keep asking different design questions, and we finally came up with this polymer. This is a polyanhydride. It's a copolymer with two units, one and the other. It's very hydrophobic, which was one of the key design requirements, because if you want surface erosion, you want to keep the water out. But you could even adjust how fast it comes in by adjusting the ratios of these. So we made these, and what was exciting is by changing the ratio of those, not only did we come close to surface erosion, but by changing those two ratios, like this case with 79% of one of the units, the polymer dissolves in 2 weeks. but pick another ratio or another ratio. This will actually last for 4 years. So you could dial in different ratios and make these last for different lengths of time. So in 1985, I was visited by a young neurosurgeon, Henry Brem. Now he's actually chair of neurosurgery at Johns Hopkins. And we started talking about this, and he told me about a problem that he was having. And that is patients that he would treat as a neurosurgeon that have brain cancer. And the worst form of brain cancer is glioblastoma multiforme. It's also the most common. And regardless of how you treat it, the mean lifespan is less than a year. At that time, the only drug that was approved for treating it was this drug, BCNU. And it's a terribly toxic drug. As many chemotherapy agents, you inject it into the bloodstream, it travels throughout the bloodstream, and it can cause very serious side effects. And what he and I talked about is maybe we could change that paradigm too. Maybe we could do something like this. What if we could introduce what we'll now call local chemotherapy? Rather than the neurosurgeon, well, the neurosurgeon's anyhow going to do surgery. But maybe when he does the surgery or she does the surgery, they could leave behind a little wafer that would have a polymer containing the drug that could locally deliver it to the tumor. Now, BCNU normally has a lifetime of just 12 minutes. But if it's put in a polymer, it's protected from degradation. It could actually last as long as the polymer lasts. And so neurosurgeons at Hopkins wanted it to last for a month. Not only that, maybe you could even get local chemotherapy, high concentrations in the brain where you want it to be, but low concentrations in the rest of the body where it might cause harm. Well, whenever you come up with an idea like this in the United States, probably here too, you have to raise money. And the way we raise money is you generally write research grants, and they go to, say, the National Institutes of Health or something like that, and they have what are called study sections. Study sections are professors at other universities, and they review these, and they tell you how you did. And we did terribly. I entitle this slide, "This Approach Will Not Work Because." So in 1981, well, when we first wrote the grant, the reviewers said the polymers can't be synthesized. But I had a very good graduate student, Howie Rosen. He later became president of Alza, a $12 billion company that I mentioned before. He synthesized them. He also became elected to the National Academy of Engineering, one of the highest honors of the United States. So then we sent it back, and the reviewers said, well, but the polymers have these anhydride bonds. They'll react with whatever you put in. But I had another couple postdocs, Bob Linhart, who's now Constellation Professor at RPI, and Cam Leung, who's a chaired professor at Columbia, also been elected to the National Academy of Engineering, and they showed that, that there was no reaction. Sent it back again, the reviewer said, well, yeah, that's okay or good, but the polymers are what are called low molecular weight. They're fragile, they'll break. I had another couple postdocs, Avi Dome, who became chairman of medicinal chemistry at Hebrew University, and Edith Mathiowicz, who's now a full professor at Brown University, was elected to the National Academy of Inventors. And they made polymers very strong, high molecular weight, which clearly didn't break. We sent it back again. The reviewer said, but we still shouldn't fund it because new materials in a person, that's certainly going to be toxic. But I had another graduate student, Cato Lorenzen. He later became Dean of Medicine at the University of Connecticut. In fact, last year he received the National Medal of Technology from President Obama. He's also been elected to several national academies, and he showed that didn't happen. Sent it back again, and the reviewer said, but the drug will not diffuse far enough to kill the tumor. But I had another graduate student, Mark Saltzman. Actually, Mark became Yale's University's first chair of biomedical engineering, which he still is today. Been elected to the National Academy of Medicine. He showed that it did diffuse far enough. Anyhow, this kept going on until 1996 when the FDA approved this for human use. It was actually the first time in over 20 years they approved a new treatment for brain cancer, first time they ever approved this idea of local chemotherapy. You can probably tell from the way I'm speaking, I'm very, very proud of how well all the MIT graduate students and postdocs did. They received all kinds of honors, fantastic jobs like deans, chair people, and received medals from the president, or one became CEO of a multi-billion-dollar company, whereas the reviewers at the other colleges, they haven't done so well. I want to show you what this operation looks like. If anybody's squeamish again and doesn't like the sight of blood, don't look. But here's a picture of these going into the brain. Usually they put 6 or 7 in and close them. By the way, I should say it's very hard to get good advice when I give a talk, but about 14 years ago, I was giving a lecture at MIT, and my wife came to the lecture, and I said to her at the end, I said, what did you think? And she said, well, Bob, she said, the lecture was okay. That's very high praise. But she said, you know, I don't know if you were aware, but you had those 2 bloody slides on for 10 minutes, and all those chemical engineers were turning green. So ever after that, I did what I did today. I showed them very quick. I gave a warning. But I just want to tell you the sequel to that story, which was that 2 years later, I got asked to give this dinner speech. This was to a group of neurosurgeons. And I told them the same thing. And at the end of this dinner speech, quite a few neurosurgeons came up to me and they said, you know those 2 bloody slides you just showed? I said, yes. They said, those were fine. They said, no problem. You could have left those on as long as you wanted. They said, but those chemical formulas. You know, to leave those on for a neurosurgeon, I guess, is very bad. Anyhow, just to show you some of the clinical data, this is one of the early papers published by clinicians on this, showing that, and it's not a cure by any means, but it does show that you can prolong survival, in this case, 5 times. But what's happened now, this has actually now been approved by regulatory authorities in over 30 countries. It's been used for the last 21 years. So now there have been papers summarizing some of the different clinical trials, and this is actually what you see with thousands of patients. So again, not a cure, but it provided a whole new treatment which relieves suffering and would prolong life on the average. Also, it created a brand new paradigm of this local chemotherapy using polymers. And not only is it used now in cancer, but actually one of my other former graduate students is now a well-known professor, Elazar Edelman. He's also a cardiologist. You know, and as well as several companies began to use this in other areas, like in cardiology. And in cardiology, one of the biggest problems today, one of the main treatments today, is if somebody has heart disease, very often they put in what's called a stent. And a stent, it looks like a Chinese finger puzzle. It keeps the blood vessel open. Problem is, half the time, within just a few months, you get a problem called restenosis. The smooth muscle cells start to proliferate, closes off the blood vessels. The person may die, or at minimum, you'll have to reoperate. And now, there's over 1 million operations a year where people put in what's called drug-eluting stents. It's actually the very same thing we're talking about. You coat these stents with a polymer, and now you put in a different anti-cancer drug, in this case Taxol, locally delivers the drug and keeps the vessels open. And there are probably other things as well that one could do. I thought I'd spend the last few minutes talking about not only how you could deliver drugs, but deliver cells with material. Let me motivate this by showing a child, and he's being treated by my collaborator, Jay Vacanti, who's a surgeon, and he's dying of liver failure. And the only way a child like this was 1983. The only way a child like that would live then or now is somebody else dies and they do a transplant. And Jay started talking to me about this, and we came up with an idea. This is from a paper we wrote in Science. It's been cited over 6,000 times. But basically, our idea is the following, is that we could take literally any cell. We published this even before people talked about stem cells, and you could use those too, as I'll go over. But basically, you take any cell type. This is bone, cartilage, liver, intestine. If you inject them at random, not much happens. But cells are smart. People have shown, like at Berkeley, that you could take mammary epithelial cells. If you put them close enough together, they can actually make what are called acini and make milk. So what we did is we want to provide cues. We actually could create scaffolds. That was our idea in 3 dimensions. And the idea is you could put cells on them, and if you grow them under the right tissue culture techniques, and that's not only involves figuring out the right media, but very often the right stresses, then our theory was that you might be able to make these new tissues. And if you could do that, there would be multiple applications. Not only, which our main interest was helping people like that baby, but it might provide a way even of testing new drugs, because you'd have artificial tissues. Maybe you wouldn't even need to use animals someday. So at any rate, just to show you some of what we did, here are some polymer fibers made out of either polymers that would exist or brand new ones that we'd synthesize. And basically, they're in 3 dimensions, and you'd put cells on them. But you could take this even further. Prasad Shastri, who was one of my postdocs, now runs a big institute in Germany, actually began using CAD-CAM techniques, computer-aided design, followed by techniques like foaming or 3-dimensional printing. And I'm just going to speculate with you where this might someday go. Let's say, here's, for example, to make a nose. let's say 40 years from now, somebody comes to a plastic surgeon and they don't like their nose. They said, I'd like a new nose. And the plastic surgeon will have a computer screen and he'll show pictures of like 50 different noses. To that end, what he was able to do is he made like a regular nose. This is 98% porous, and then you could even put cells on it. But let's say somebody wanted not just a regular nose, let's say they wanted an upturned nose. Well, that wouldn't be so hard. We'd take a little bit of this off. Let's say they wanted a hook nose. I mean, they probably wouldn't, but if they did, we'd give them a little bit more. And so you could, make the tissues like this. I thought I'd show you 3 quick examples to illustrate this. So first, let's say you wanted to make new cartilage. Like I just showed you, here we redid this guy's skull. These are nude mice. By the way, here you shouldn't get rejection because you're using the animal's own cells, and that might happen for the person as well. Here we redid this guy's cheek. If you open the animals up and look at the tissue, it's pure white glistening cartilage. Histologically, it looks like cartilage too, but it turns out it does not have the mechanical properties. If some of you are runners, we're not able to fix you yet. But you can do cosmetic things. So the Army actually approached Jay, myself, and Linda Griffith, one of my former postdocs, because they see people who came back from Iraq or Afghanistan without body parts, like new ears. Could we make an ear? Linda actually made a scaffold of a human ear. Here's a high-powered scanning electron micrograph of that with the cells and making matrix. Here are the polymer fibers. These will ultimately disappear. They'll break down to water and carbon dioxide, and you'll get all new tissue. And this has not yet been put on patients yet, but it has been put on animals just to prove that it's safe. Here's a rabbit with a human ear. And the reason it's done on animals is because you want to show that it's safe for humans. Even though it hasn't been done on the soldiers, it has been done on patients. Here's a 12-year-old boy. If you look at him closely, notice he's got no chest covering his heart. But like other 12-year-old boys, he likes to, in the US, he likes to play baseball. So you can imagine if he ever got hit in the chest with a baseball, he could die. So Jay operated on him, and we made a polymer scaffold for him, took his own cells and made him a new chest, and now he's doing fine. We've also licensed it to companies to make new skin for burn victims. Here's a 2-year-old boy, very badly burned. But you can take the product, which is neonatal skin fibroblasts on a polymer scaffold. You can put it in the child at the time of injury. If you come back, though, 3 weeks later, he's doing better. 6 months later, he's pretty much healed. These are now approved by regulatory authorities for patients who are burned and patients who have diabetic skin ulcers. And one final example to close this talk, I thought I'd go over work of Erin Levick, who is one of our graduate students, now professor at the University of Maryland. Erin was interested in treating people who who were paralyzed. Could we actually make a new spinal cord someday? And she was a material science major, so she made a scaffold that might mimic the white matter and gray matter of a spinal cord. An outer part which might provide axonal guidance, and an inner part which we might seed with neuronal stem cells. Here are some pictures. This is what the scaffold looks like macroscopically, but if you do a high-powered scanning electron micrograph, you see pores, and through these pores, we might even add neuronal stem cells. And here's the outer part, which through a sublimation technique, she created this nano pattern to guide axons. Here's the animal model that Ted Tang and Evan Snyder, who we worked with, created. And basically what they did is they would take out a 4-millimeter hemisection of the spinal cord. And in its place, they would do one of 4 things. We had an N of 13 for each. In one case, did nothing, sham operation. In another case, cells by themselves. In a third case, polymer by itself. And the fourth case, the polymer cell combination. They put these implants in within 24 hours of the injury and would follow the animals for 400 days. Let me just show you videos at 100 days of representative animals. This has all been published, but here's a sham operation, and you'd get the same with cells of an animal. And this is the mean of the control group. And notice that he's not able to move his hind legs very well or support his own weight. Notice also the paws are splayed in an awkward way. There's actually what's called the BBB scoring system, which goes from 0 to 20, 20 being what people here would have. He gets a 5. But if you put the scaffold by itself in, interestingly, you get a significant improvement, maybe up to an 11. And if you put the scaffold and the cells in, you go up to 14, and here's that on the average, also at 100 days. This animal is, even though he's kind of heavy, is able to support his own weight. And notice the paws are splayed much more normally. I'll show you that a little bit more in a second. But you kind of get the idea. Here's the paws. My wife tells me not to leave that on too long either, but I will leave it on for a second to just tell you where we are. So we repeated this on non-human primates, and then we actually started a company, licensed this again, and we started, they actually now have gotten approval for human clinical trials. It's being done in 31 hospitals around the United States and Canada. Let me just show you the first patient. And by the way, they're just, because of the FDA issues, decided to initially just use the scaffold by itself because there was some improvement rather than put the scaffold in stem cells, and even though that works better. But let me just show you what happens. Here's the first patient who was a 25-year-old young man in Arizona on a motorcycle accident, became paralyzed. Here he is 3 months later in a swimming pool with his sister. And basically, it's largely a safety trial, but there are no adverse events. Here he is. He has active movements of his hip flexors, palpable contractions of his knee extensors. Initially, he had no bowel function. That's been regained. Initially, no bladder function. That's been improved.
That is amazing.
Here he is at 9 months. And what they've done, this is going to be the final slide that I'm going to show you, is basically now I think a number of patients have been enrolled. They're trying to do 20 patients overall. And there's, the scoring system they use for humans is what's called an ASIA scoring system. And they look for some improvement, which could do, ASIA A is, really no good bowel function or ability to move your legs. And you could get conversion to ASIA B, ASIA C, ASIA D, ASIA E. ASIA E would be, again, like people here. He's actually ASIA C, but at this point. But what's happened, and this is the final slide, is if you look at, and it's early, but basically, according to the FDA, if you could get over 25%, they'll approve it. Normally, if after 6 or 12 months, you get 15%. So far, they've gotten 62.5%. They have to follow the patients over time. Sometimes people could even revert. But the thing is so far, it's off to an extremely good start. And we'll see what happens. In conclusion, I should point out we're also working on many other tissues. We're working on new vocal cords, new spinal cords, heart muscles, a new intestine, many others. Some, like I say, are for in vitro testing systems. Some are actually to help patients, and we want to see all these things. And some is just basic research to understand the interaction between materials and cells, or to create brand new materials, because these are all needed to try to solve these problems. So in summary, what I've tried to go over today is really go over a little bit of my personal journey from being a chemical engineer who might have worked in the oil industry to what we're doing now. And to really try to illustrate some of the struggles that we've had, but at the same time illustrate some of the challenges, and the hope that by doing this kind of work that you can relieve suffering and prolong life. Thank you very, very much for having me. Happy to. Of course, thank you. My pleasure, thank you.
Okay, so thank you, Bob, for a truly inspirational talk. I'd like to open the floor to questions. There's a few mics going around, so if anyone has a question, can you raise your hand, and one of the PhD students will, I think there's a mic going around, is there? Yeah. So maybe just over here.
Thank you very much for a very inspiring talk. I have a question about a specific area that I'm particularly passionate about, is the use of nanomedicine to overcome the blood-brain barrier.
Sure.
And that's a very tough area, because the drugs most of the drugs, they don't cross the blood-brain barrier, but nanoparticles with drugs, they don't do either. So what would be your vision? Will it be how will nanoparticles, nanomedicines, go through? What would be the chemical methods to initiate transcytosis, for example? What would be physical? And beyond the blood-brain barrier, will it be the small molecular drugs that will diffuse all over the place? Or you also mentioned the next generation biotech drugs, the siRNA or messenger RNA, that have to be inside the cells. But in that case, how will they cross the extracellular matrix?
Yeah.
So what is your vision for that area?
Right. Well, so the question, just to go over, is for people that aren't familiar, trying to deliver drugs to the brain has been particularly challenging. And there's something called a blood-brain barrier, which is a tightly packed endothelial cell sheath that stops molecules from getting through it. And that has been a huge problem in medicine. And for any of you that, for example, have followed new treatments for Alzheimer's, a lot of people think that one of the reasons why every single trial has failed is because of what you just said, that those molecules don't get through the blood-brain barrier. I mean, even if they work and could stop amyloid or whatever else, if they don't get there, how can you do it? So, I mean, it's a great question, and I wish I could tell you that I had a great answer. I mean, so sometimes you can do it because the blood-brain barrier is compromised. In the case of the brain cancer that I talked about, there we actually did implants. Some of the strategies, whether it's nanoparticles or not, I mean, that people, including ourselves, have looked at some, are sometimes doing heroic things. Could you disrupt the blood-brain barrier by using techniques like ultrasound? Could you create new particles? For example, there's some evidence that exosomes, which are part of a cell, might be able to cross the blood-brain barrier better. And maybe if the way I would probably go about it is to do more basic science, to try to understand if I had something like an exosome, if that truly could cross the blood-brain barrier, to try to understand, almost take it apart step by step and find out what is the physical chemistry or molecular chemistry that enables that to happen. I just think there's such a poor understanding of that. That's further complicated, in my opinion, by the fact that I don't think there's very good models for studying it. In other words, what mimics the human blood-brain barrier? One of the problems is that, if you use small animals, there have been reports of some bioavailability across, small animal blood-brain barriers, but I don't know that there's good evidence that that mimics what happens in the human blood-brain barrier. So people are trying to create organoids, again, organs on a chip or things like that, to try to study it. But I don't know that those have recapitulated what actually happens in a human either. So I actually think there needs to be a lot of work. I wish there was the funding for all of it to, A, create better models, both in vitro and in vivo, B, to get a more fundamental understanding of what causes transport, and again, there are transporter systems and things like that, and C, to create new materials based on that that could traverse everything. But I think probably all those things will need to be done.
Do we have any other questions? I'm sure there's a few. Yep. Edmund?
Thanks very much. That was a really inspiring talk. I have more of a general question. So you described very eloquently how you didn't get grant funding and how you were told that this wouldn't work. But as you may be aware, there's a huge impetus on targeted funding.
I'm sorry, what?
There's a huge impetus on targeted funding.
Yes.
That funding for research has to be targeted into areas A, B, C, and D.
Right.
And there's a danger then that you may not actually develop new things that actually won't work. And I'd be interested to hear your opinion on funding.
Yeah,
in those
well,
kind of
actually, let me make a couple points on that, because so the issue is on funding, and probably, you might ask if I didn't get funding on the NIH grant over and over again. I'm glad you reminded me of that. How did we ever do this? So, and I should tell you what I did is I kind of bootlegged in the beginning from the grants I did have, but that still wasn't that much money. So actually, one of the ways we got funding is we wrote patents, actually. And even though we didn't get funding for a long time on this, the patents, we actually got a company to license it, and they gave us several $1 million, because that was sort of the quid pro quo. And they actually got to sell those systems, and everybody won on that. Interestingly, just to close this, it relates a little bit to your question, is after we got FDA approval on this and never got funding from the government, then they had what's called an RFP, a request for proposals, to put better drugs in the same systems we developed. which is kind of typical of what happens. But the point that you're asking is about targeted funding. And I personally feel the most important thing that you could get if you're a scientist at any stage is untargeted funding. There's things to do really blue sky research where you don't know where it's going. And even though I'm an engineer and all the things I showed you today are engineering, if you really look back at what I showed, when we started delivering macromolecules, when we created these new polymers, when we created the microchips, when we created the tissue engineering, we didn't have a clue about where their applications would be. I had the belief that it would open up new areas, but the fact is the reason those new areas happened wasn't necessarily because of me. It's like we created technologies that didn't exist before, but it were clinicians who came to us and said, well, maybe you could use it for this, or maybe you could use it for that. And I think it's really through that combination of almost doing non-targeted work in the beginning that those technologies got created. So I'm a big believer in basic science and engineering research. And I think that I would say today, the more we can do to allow people to use creativity, to explore their curiosity, I think is a good thing. That doesn't mean there shouldn't be targeted research either. I think that can be a good thing. And I think that can often happen, like say in our case, I didn't get into this in the talk, but we've had targeted funding sometimes from foundations. I mentioned tissue engineering at the end. So about 10 years ago, the Juvenile Diabetes Foundation came to me and they said, can we help them come up with new treatments for that with tissue engineering? And they've given us about $20 million to do that. And we published actually in the last 2 years 4 papers in Nature going over how one might create new material cell combinations to do just that. And I think that's good too. In other words, but there are foundations, there are companies that will often give you targeted research. So I think trying to get untargeted money to me is precious. So I think that I certainly am in favor of that if it possibly could be done. Any other questions? Well, it's okay. You don't. That's my. Anyhow, thank you very, very much. Pleasure.
Just as an appreciation of your time and your visit here, you've met and spoken with a lot of our PhD students and other investigators. So it's just a small token to remind you of Limerick when you're back in Boston.
Thank you very much. Okay, thank you. Thank you very much.
Okay, I think it's time for lunch.
Okay, sounds good. Thank you.